Hello everyone. This is Dr. V. Aruna, Assistant Professor, PG and Research Department of Microbiology, Cauvery College for Women. Now we discuss about mutation. Mutation is the change in sequence of nucleotide of DNA. Change in sequence of nucleotide brings sudden change in morphological characters of an organism. If such change are heritable, then it is called as mutation. So mutation is defined as any heritable change in the sequence of nucleotide of DNA. Organism with mutation is called mutant, while the organism without mutation is wild type. Mutations can be bad, lead to cancer, aging, birth defects, self-aborted embryos. Types of mutation. Based on change in genotype and phenotype, mutation are of two types: point mutation, frame shift mutation. Types of gene mutation. First one is point mutation. This is subdivided into three: missense, nonsense, and silent. Frame shift is subdivided into two types: insertions, deletions. Point mutation is a genetic mutation where a single nucleotide base is changed. Missense mutation is a change in one DNA base pair that result in the substitution of one amino acid for another in the protein made by a gene, for example, single cell anemia. Nonsense mutation. A nonsense mutation is also change in one DNA base pair instead of substituting one amino acid for another. However, the altered DNA sequence prematurely signals to the cell to stop building the protein. This type of mutation results in a shortened protein that may function improperly or not at all. Example include thalassemia. This is a normal condition of gene sequence. This is a altered gene sequence. Silent mutations. Silent mutations are mutations in DNA that do not have any observable effect on the organism's phenotype. For example, if the codon AAA is altered to become AAG, the same amino acid lysine will be incorporated into the peptide chain. Frame shift mutations. Insertion mutations. An insertion changes the number of DNA bases in a gene type, adding a piece of DNA. As a result, the protein made by the gene may not function properly. Deletion. Deletion changes the number of DNA bases by removing a piece of DNA. Small deletion may remove one or a few base pairs within a gene, while longer deletion can remove an entire gene or several neighboring genes. The deleted DNA may alter the function of the resulting proteins. Chromosomal mutation. Chromosomal mutation may affect one or more than genes. For example, non-disinjection translocation. Somatic or germline mutations. In this case, the embryo will carry the mutation without any of its parents or posed in his genetic heritage. This type of mutation occurs during the formation of life or the gametes of one parent. That's all about the mutation. Thank you, everyone.